Okay. Hello, everybody. My name is Sophia Bagley. I'm a senior at the Harker School, and I'm part of the new lab.
Hi everyone, I'm Jacqueline Shane. I'm a senior at the King's Academy and I'm also part of the new lab.
Today, we'll be discussing our scalable formal synthesis of R plus Etomoxir without pyrophoric reagents enabled by benchtop NMR. We will first go over the background of the CBT system and discuss our previous work on Etomoxir and discuss our current synthesis, including our oxidation optimization and a newer offshoot project for Mosher's esters, and then we'll conclude with our future plans for this project. So in order to discuss the CBT system, we must first look at the mitochondria, which take in fatty acids as fuel for generating cellular energy, where lipids are transported into the mitochondrial matrix, converted into ATP through a process known as fatty acid oxidation. The CBT system contains 3 transmembrane enzymes found on the outer mitochondrial membrane. These proteins bind fatty acids to carnitine, an amino acid naturally found in the CBT system, forming a complex that can effectively cross the membrane. This system has been identified as a target for treating dysfunction in fatty acid metabolism, particularly in diabetic biological systems where sluggish glucose catabolism leads to an accelerated lipid biosynthesis. Its metabolic pathway has major implications in diabetes, COPD, asthma, as well as cancer. CPT1 in particular is a rate-limiting mitochondrial enzyme that supports fatty acids into mitochondria for beta-oxidation. It is a great pharmaceutical target for the aforementioned diseases. And inhibition of CPT1 reduces long-chain fatty acid transport into the mitochondria, which raises whole body glucose tolerance by shifting cellular energy production to glucose oxidation and increasing insulin sensitivities. So our project addresses the synthesis of Etomoxir that has a similar structure and function to carnitine, seen in the bottom right corner of the slideshow, where an esterase makes a nucleophilic attack. Next slide, please. So more on Etomoxir. Etomoxir is one of the most well-known inhibitors of CPT1. So it has 3 structural components: an aryl ring, which is highlighted in blue here, which pi stacks to a key histamine residue within the binding pocket. And then our middle portion is the carbon chain in green, which allow for hydrophobic properties similar to the fatty acids that CPT1 usually catalyzes. And it also keeps it in place with the binding pocket. And lastly, we have the alkylating group in red, which irreversibly and covalently bonds to a key cysteine residue within the CPT1 protein complex. So Etomoxir has gone through phase 2 trials and showed good promise. It has showed effective irreversible inhibition of CPT1A, which is an isoform of CPT1, and it has also shown ventricular hypertrophy prevention of CPT1B. However, its lack of selectivity between CPT1A and CPT1B ultimately resulted in its removal from FDA phase 2 clinical trials. And in this clinical trial from FDA phase 2, it was meant to treat heart disease. So the Etomoxir would target CPT1B and it would be an attempt to inhibit CPT1B. Unfortunately, it also inhibited CPT1A, which is the isoform associated with the liver. So these patients ended up getting induced hepatotoxicity or chemical-driven liver damage. Thus, the clinical trial was canceled prematurely. So previously, in other Etomoxir-related studies, 2,4-dinitrophenol was previously synthesized as a molecule that showed lower toxicity and higher selectivity. Yeah, it was like very similar to the previous Etomoxir molecule. Therefore, we conclude that the aryl ring is used as an addition to the Etomoxir molecule to create more selective analogs. And We hope that by synthesizing a library of Etomoxir analogs with aryl fragments of different properties, like hydrophobicity or electronegativity, we will form more selective analogs between the two isoforms. In a previous synthesis of Etomoxir in Auerbach 2004, the 2,4-dinitrophenyl analog shown here at compound 1.7 was synthesized in a 6-step synthesis with approximately a 22% overall yield. Unfortunately, there is one major step that took about 16 to 18 hours, depending on the scale, which was inefficient and not manageable for a multi-step synthesis reaction. This step is a homologation using TMED methyl alcohol, but most dangerously, it used n-butyllithium. So I'll go more in depth about what n-butyllithium is and why it is such a downside to this reaction. So n-butyllithium is a dangerous pyrophoric agent with a deadly history, and in order to use something like a pyrophoric substance, one must use a long syringe, fireproof lab coats, and reduce unnecessary air or moisture getting in. Seen in the diagram on the slide, it is a very rigorous, very difficult reaction, and there is a high likelihood of danger. So if a needle cracks or a syringe breaks, it becomes extremely dangerous. And our job here in this project is to reduce this danger. Furthermore, yield was only 66%, so it wasn't that effective of a reaction.
So, taking into the downsides and the dangers of the previous synthesis that is reported, we came up with a new retrosynthetic pathway for synthesizing the R enantiomer of Etomoxir. And so, as you can see here, we start with commercially available octane-diol, which is compound 2.0, and then we desymmetrically monosylate it with TIPS trifolate to create compound 2.1. And then we oxidize this monosylated compound to make the aldehyde. And then we form the aldol, which is compound 2.3, through a aldol Luce reduction. And then we perform the sharpest epoxidation to create compound 2.4. And I want to direct your attention to compound 1.4 here. It's a very special and important compound because it's a very diversifiable intermediate, which means that we can easily modify this compound to create a myriad of different analogs, like the analogs that Sophia talked about earlier. If you look at this step, which is where we form the aldol, this step reduces the need for n-butyllithium, tert-butyllithium, which makes this reaction a lot more scalable, and it removes the pyrophoric reagent, which also removes the need for an 18-hour long extremely rigorous reaction. And so one of our first challenges was figuring out how to oxidize compound 2.1 into compound 2.2. So we tried 11 different oxidation conditions, as you can see on the table on the left, and our most yielding condition was our last entry, which is entry number 11, where we used a copper catalyst, bipyridine, and methylimidazole, tempo, and oxygen as an oxidant, where we were able to achieve a yield of 84%. And we determined this by tracking the reaction using a 1H NMR. This is the copper cycle for the copper catalyst reaction, entry number 11. And the reason why this reaction is so nice is because, as you can see, it forms a catalytic cycle, which means that we begin with copper 1, which is our copper MECN4PF6. That's our copper catalyst, and copper is in its +1 state. And then after reacting with oxygen, it becomes a +2 state, and then it reacts with a bunch of other stuff, and then it immediately goes back to copper 1 after it oxidizes our TIPS monosylated material. And so what's really nice about this is that we don't need a large amount of copper catalyst in order to make this reaction happen. A little bit of copper catalyst goes a really long way. So we only use around 2.5 to 5 mole %. in order to actually carry out this oxidation. And another really nice thing is that we're literally using oxygen as our oxidant. Also, we're able to track this reaction visibly because in its resting state, or as copper is in its +1 state, the reaction is a burgundy reddish brown color. But then after it becomes copper 2 plus, or when our reaction is almost complete, the reaction will turn to a greenish turquoise color. And so it's really easy to visibly determine when our reaction is done. Another challenge that we had was, as you can see on when we go from our aldol to the epoxide, there's two possible stereochemistries. There's the R enantiomer, which is here on the top, and the S enantiomer, which is on the bottom here. And so since we're forming the R enantiomer of Etomoxir, we only want the R enantiomer of the epoxide. And so we've developed a method to use Mosher's acid chloride analysis in order to determine which enantiomer we have. And so we attach the S enantiomer of the Mosher's acid chloride, although we could also use the R one, but here we just chose S. And we should be able to tell the differences between the two enantiomers based on their 19F fluorine signals. So to make sure that that actually works, we've tried it with a bunch of different compounds like R-sec-butanol, S-sec-butanol, plus menthol, and L-menthol, and we attached both enantiomers of the Mosher's acid chloride. And so we formed four different compounds for each compound that we test. And then we put these all into 1H NMRs and 19F NMRs to determine whether or not there's a difference or a shift in the peaks. And we noticed that notably for the R-sec-butanol attached to the R Mosher's chloride or attached to the S Mosher's acid chloride, it has a 0.1 ppm shift. And so that shows that yes, we can actually distinguish enantiomers based on Mosher's acid analysis. For our future plans, we need to finish making our analog library shown on the left, and then put all of these analogs into cells to determine the selectivity and efficacy of our compounds and determine if they actually do inhibit CPT1. And we also need to optimize our formal route synthesis. Thank you to the rest of our group members, Rushika, Elizabeth, Anka, and Sarah, to our advisor, Dr. Edward New, and our collaborator, Dr. Chen, and of course, ASDRP. And here's a QR code of the previous paper that New Lab published regarding CPT1.